Hello, I'm Ny'Kera Hall, and I'll be pleased to present my research of the development of multiplex PCR primers for the detections of incognita, H. glycines, and R. reniformis in soil metagenomic pool. This research was supported by South Carolina INBRE, Converse College, and Clemson University. Plant-parasitic nematodes are multicellular roundworms that invade and feed off of plants by inserting themselves into the plant root and burying their head inside, creating the host and feeding off different nutrients from the plant and for themselves. This has caused a major concern for scientists and farmers, as it has caused these nematodes have caused approximately $80 billion in damages annually. These microscopic roundworms are really a major concern because as when they deprive the plants of their nutrients, it basically drives the plants away from the products farmer needs that provides us with daily needs, for example, food, fabric, etc. The three nematodes that are economically important to South Carolina are the southern root knot, which is Melodogyn incognita, the soybean cyst, heteroderic glycines, and reniform nematode, which is Reniformis reniformis. Scientists seek to identify the nematodes in soil management pools so they would be easier to detect to prevent any harm done to the plants before it's too late. Multiplex PCR was produced in this research because of its advanced enabling of simultaneous amplifications of many target regions in one reaction with more than one organism and paraprimers. This will help determine if nematodes are present in the research to detect and identify each based on their primers. And the hypothesis goal of the study was an aim to produce multiplex PCR primers to identify these soils and soil management pools using DNA sequences selected from GenBank. During the development of this research, methods have, as shown, have been modified to produce better accuracy and detection for nematodes and multiplex PCR. Previously developed primers for these nematodes were combined in multiplex polymerase chain reaction. The original progress was done by literature search in which many articles of researchers that have done a similar research compared to detecting nematodes were found in which they picked the RNA sequence of the nematodes for their primers. These were picked and these were found to detect if any of these would give a good match to the nematodes based on their base pair and also their primer sequences. A temperature range of 55 to 60 degrees Celsius was used to distinguish the acceptable temperature of melting for annealing when it comes to the annealing of the PCR for the nematodes. This was thought as a good temperature range because it would have gave a better accuracy and a better detection. When the PCR is ran and goes through gel application, it would give a better better chance at detecting the nematodes based on their DNA bands. The concentration for the dilution of the primer mix and the master mix was adapted. Over time, there were concentrations that was used to use in the primer mix and master mix for the nematodes for each. Each would usually work itself. There was always a problem with running leakless nematodes and many modifications was done to find any difference to the nematode DNA band when found in the gel. 200 microliters of each nematode were centrifuged for 20 minutes at 10,000 xg for DNA extraction. This process was done to create a centrifuge to where the majority of pure nematodes were at the bottom, so the supernatant was removed to insert DNA extraction, so the DNA would be extracted before being run on PCR with primer mix and master mix. The PCR was run under the following parameters: an initial denaturation step at 94 degrees Celsius for 5 minutes, followed by 5 cycles of 94, 45, and 72 degrees for 30 seconds and 1 minute respectively. A final extinction was carried out at 72 degrees Celsius for 7 minutes. Overall, this PCR would usually run on average 2 hours each time it ran. The parameters were adjusted to promote optimal amplification, as amplicons were run on a 2% TBE gel to determine PCR efficiency. and the gel application was performed for under 60 volts for 90 minutes to give time and more for DNA bands to be produced under amplification. This first result shown here was when gel was run using actual whole nematodes. Our first method was trying to see what whole nematodes be shown in the in the gels to show any signs of bands from just whole nematodes, as well as 1 through 8 are shown to be prominent, pretty faint. However, these are only DNA ladders that were inserted into the gels. As for whereas 2 through 7, these would have represented our samples: Melodagine, Heteroderic, and Reniformis reniformis nematodes. So the first result was from our original methods that we had tried to develop that unfortunately gave a few errors that we would presume to be from any PCR inhibition, any materials that were affecting our results, and etc. So we decided to use the modified way of our methods to see any results for our second time of trying for different detections for the nematodes and PCR. This second data result shows the modified method that was used in the research. However, with this set of results, it was split into two due to one part of the materials being used was older primer batch and new primer batch sets from each nematodes that were made when PCR was produced and gel application was done to see any results of DNA bands from the nematodes. And thankfully for both with the modified method, there was actually a very good sign of DNA bands being shown in both sets of the gels. However, with the original batch set, it was a bit more clear to see, even though it's very faint from where I'm pointing it. There were signs of DNA bands for each nematodes. However, running leakulus, as shown when compared to Melodagine and Heterodera, it was still pretty faint, and that's a complication as to doing the research. We're trying to develop and figure out why is it that running leakulus is the only nematode that's not producing right or showing good DNA band results when compared to Heterodera and Melodagine. And along with the new primer batch, however, as pointed here, you probably may not be able to see it much, but the DNA bands on here, however, are very, very faint when compared to the original primer batch. So it may be in between. Maybe there's a difference with the original compared to the new primer batch as to why there's a difference in the results. Maybe we're trying to figure out is maybe it's a difference with the reactions with the methods being used or the materials being in contact. We are still trying to figure out that process to get better and looking results for DNA bands in the gel. So based on the results given during this research and the progress, we're still figuring out that there may be a complication in the research that still needs to be discovered. And two possibilities are PCR inhibitors, which there could be something in the materials, or maybe a different method how to run the PCR to give it more accuracy and more detecting for the nematodes. That's one issue we may have to discover during the research, or the primer concentration optimization that we still need to figure out what's the right concentration for each primers that will be used to help when it comes to running multiplex PCR in the future, detecting the nematodes and the DNA bands in the gel, and along with detecting them in the soil when the time comes for the time for it to detect them in Soil Metagenomic Pool. And the group will continue to work on optimizing this reaction and to hopefully find the results needed for this research to detect nematodes and soil metagenomic pool. Thank you.